So did you have this stuff already? I think we did that already.
Oh, like, no.
Oh, okay. Oh. It's good. I didn't look at it, but I'm still trying.
Oh, you
That's what my dog is taking for
Yes. Okay.
Yeah, 5.
Thanks.
As soon as it gets that high, it's cool. Is that all? I'm just wondering. I got it. On the
Okay. Yeah, I was a Tuesday.
Okay, folks, we're gonna go ahead and get started. So, this first thing I need to do is press this on. So as of this morning, I was able to get everything on Blackboard organized, so it should come up correct for this week and next week now. I also went into Blackboard and added the PowerPoint for 18 and 19, and then next week you'll see 20 come up. As far as the exam is concerned, I expect it to cover chapters 18 and 19. The other thing I did is there was a homework according to Blackboard that was originally due Monday. And there was a quiz that was due Tuesday. Both of those you were given your points. So you got 2.5 for the homework and 10 for the quiz. And I just uploaded those grades. So those are some of the things that I've done this morning in preparation for class. What we're going to be doing today is we're going to finish up chapter 18 on proteins, and then we're going to get into chapter 19, which talks about enzymes. And it's our main focus in chapter 19 will be that about enzymes, even though I believe it's enzymes, vitamins, and something else. So let's pick up where we left off on Tuesday. So on Tuesday, we took the amino acids, we combined the amino acids to make primary structures. Primary structures are simply the order the amino acids are combined in. We took and talked about the fact that from the primary structure, you can develop the secondary structure. The secondary structure is really what occurs along the backbone. So is it a beta sheet, meaning that along the backbone, they're basically nice and in a straight line, and so the backbone readily hydrogen bond? Is it in an alpha helix, meaning every fourth protein hydrogen bonds to the fourth one down? Or is it a globular protein, which means that there is some hydrogen bonding among the backbone, but it is less so. We then talked about the tertiary structure. With the tertiary structure, that's where we really start looking at the side chains and what the side chains do. Do they make a non-polar pocket? Do the side chains come in contact so that they can hydrogen bond? Do acidic and basic side chains come in contact so that they can make salt bridges? Do we get a disulfide bond? Those side chain interactions are extremely important when we start talking about how those globular proteins form their shape. And then we talked about quaternary. The quaternary is basically when you have multiple proteins come together to do a job. After that, we started talking about how you can destroy a protein. And one way to do it is by using hydrolysis. And the hydrolysis is the exact same process that we talked about in chapter 17 for the hydrolysis of an amide bond. Basically, if you talk about hydrolysis of an amide bond, you are talking about breaking the C double bond O in bond and adding water into the structure. Now, unlike the organic reactions we talked about in 17, in order for this to occur, you're going to have a catalyst in order to break the amide bonds in a protein. It doesn't just occur. Otherwise, we would have a lot of broken proteins in our bodies. So you have to have a catalyst. So when we talk about hydrolysis, again, we're talking about adding water and breaking this C double bond O NH bond. Now, so let's just look at a possible way that a question could be asked about this. An example of a catalyst that causes hydrolysis in the body is the enzyme trypsin. Trypsin hydrolyzes the peptide bonds on the carboxyl side of lysine and arginine. So it looks specifically for those amino acids, and it will break the peptide bond on the carboxyl side of those amino acids. What will the fragments be from the following sequence after the hydrolysis? Now, one thing you have to remember about what we talked about when we developed the proteins and we developed them into the primary structure is we always write them in a certain direction. When you develop the proteins, the N-terminal is always on the left-hand side, and the C-terminal is always on the right-hand side. That's important because when we talk about breaking this, it tells us that it breaks at the carboxyl group. It means it breaks at what would be the C-terminal. So, if it works on lysine and arginine, and it's going to be on the C-terminal, which is the right-hand side, then one of the bonds that's going to break is going to be the lysine-cysteine bond. And the other bond, since it breaks arginine on the carboxyl side, is going to be the bond between arginine and leucine. So our fragments that will remain are going to be alanine, phenylalanine, lysine, with the lysine being the C-terminal. We will get cysteine, glycine, asparagine, and arginine, with arginine being the C-terminal. And then we will get leucine, phenylalanine, glycine, alanine as our third fragment. Any questions about that? When you do, by the way, as we'll talk about it in the next chapter, when we talk about very specific enzymes, we're going to eventually talk about how enzymes are named. There are a few that have a common name like trypsin. And if it has a common name, it doesn't tell you what goes on, which is what the naming of enzymes typically do is it tells you what goes on. But if it does not tell you what goes on, it will be specified in the question. So in this particular case, you can't get an idea of what trypsin does. So that's why they told you that it breaks the peptide bond on the carboxyl side of these particular enzymes. I mean, not enzymes, amino acids. questions?
Are there any books in the textbook that sets it up like this that we can practice over?
Yes, I'm sure there are. So let's see. That's quaternary. So under the chemical proteins, page 588, it talks about some of these processes. Under the conceptual I'm looking for one. It's definitely on the homework more conceptual. There, the example that I just used, and there's another worked example, is on page 581. This will come up more, however, when we start talking about enzymes. So we'll talk about this idea more. Right now, it's really what is hydrolysis and understanding what hydrolysis is, which means it's breaking the amide bond.
Okay.
Okay. So there are other ways that we can destroy proteins. We talked about one of those ways, which is hydrolysis, where we're breaking some amide bonds. Another way is to denature the protein. Basically, if you think about that globular protein that I showed you, there was the nonpolar pocket, and there was the disulfide bond, and there was the salt bridge, and all of that came together to make up this nice, beautiful globular protein. If we denature a protein, what we're doing is we are messing up the structure. So we could be taking and causing the hydrogen bonds in the alpha helix to break. Or we could be going into that hydrophobic pocket and causing the hydrophobic pocket to open up and no longer be this nice closed piece. We could add in something that is going to break that salt bridge, something that positive is going to prefer to react with in the salt bridge, or something that the negative is going to prefer to react with in the salt bridge. So basically, one opportunity is to denature. And to disrupt all of those forces that come together. It doesn't mean we're hydrolyzing. We're not breaking the bonds in this particular case. Instead, we're just interrupting the intermolecular forces that are going on with the side chains. So when we start talking about denaturation, we are talking about messing up the side chains. One way to do that is heat. I mean, every time you make eggs, that's what you are doing, is you are taking the albumin and the protein within the egg, and you are denaturing that protein. Hence the reason the white turns white. is you are denaturing it. So heat is one way to denature a protein. However, there are other ways to denature a protein. Like I said, all we got to do is interrupt those intermolecular forces. We either interrupt the intermolecular force along the secondary structure, or we start to interrupt that intermolecular force along the tertiary structure. But we're interrupting those intermolecular forces. So heat is one way. Another way that we can interrupt those forces is mechanical agitation. That could be in the form of stirring. It could be in the form of just shaking it real good. There are other ways. Detergents. Detergents are an excellent way to interrupt those forces because there are two parts to a detergent. When we think about a detergent, we have a nonpolar tail and a polar head. Well, the nonpolar tail could interrupt the nonpolar pocket. That is one way that we can interrupt things, is instead of these nonpolar pockets, the side chains being able to interact, now they're interacting with the nonpolar tail instead. Same thing over here. There was a polar section. You could have a polar section of your protein. And if that polar head comes in, it can disrupt the polar section of your protein. So detergents are one way to interrupt. Organic compounds. We're going to talk about enzymes. And one of the things that enzymes do is they take in certain molecules into the active site. Well, one thing we can do is screw it up by denaturing the proteins that make up that active site. So you can get organic compounds in there. You can change the acidity and basicity of the acid-base salt bridge. You can add inorganic salt. And again, that's going to interrupt that acid-base, that positive negative salt bridge. So these are just some ways that you can denature your proteins. Basically anything that comes in and can interrupt side chain interactions or interrupt that secondary structure can denature a protein. It doesn't mean it destroys the protein, meaning it doesn't break it up like the enzyme would. It means that it destroys the protein in that it's no longer going to have the same folding, the same tertiary structure. So denaturation is about the tertiary structure. Hydrolysis is about the primary structure. Questions? Secondary and tertiary structure. Basically, it interrupts the intermolecular forces. Technically, I guess even denaturation can interrupt the quaternary, but basically it does so by screwing up the secondary and tertiary. Questions? Okay. One reason we have to talk about proteins is proteins, they make up a huge portion of the body. And one thing that they are great at is making up the enzymes. So we needed to talk about proteins so that we can understand enzymes. And we need to talk about enzymes because enzymes are responsible for catalyzing the reactions that go on within the body. So an enzyme is a catalyst. Now, I imagine that you discussed catalysts in general chemistry for health sciences. Basically, as a reminder, a catalyst does not affect where equilibrium occurs, and a catalyst cannot cause a reaction to occur that is simply energetically unfavorable. What a catalyst does is it lowers the energy of activation so that you can get to equilibrium quicker. And the primary catalyst for biological reactions is going to be that of the enzyme. So chapter 19 is going to talk about enzymes primarily. That's going to be our major focus. We're going to talk about enzymes in general. We're going to talk about the fact that some enzymes require a cofactor and what a cofactor is, or what it can be. We're then going to talk about classifications of enzymes. Enzymes can be categorized by a class and a subclass. Basically, and by doing so, it describes what that enzyme does. We're then going to eventually talk about how enzymes work to catalyze the reactions. And we're going to talk about factors that can affect enzyme activity, which includes some of these things like pH and so on and so forth. And we're going to talk about reversible, irreversible reactions, and how we can control, basically what we can do to control the enzyme. So that is a large portion of what we're going to talk about. We'll talk about vitamins very, very briefly, but in general, this chapter is all about enzymes. So enzymes, the majority of them are water-soluble globular proteins. And as we've already talked about, these globular proteins can be large and complex because they have all of these secondary intermolecular forces. They have the tertiary intermolecular forces, and all of this is making up this globular protein. So they tend to be large and very complex. Because they are large and very complex proteins, there are more ways that they can connect with reactants in the active sites, causing the reaction to speed up. And because they are very complex, we do have ways that we can control the enzyme activity by adding additional molecules into either the active site or an external site. So again, these globular proteins, they are massive. If you just simply look at it, you're like, I don't understand the shape until you get into it and start looking at those tertiary intermolecular forces. Globular proteins, what they do is they have an active site. The active site is going to consist of amino acids that can cause bond breakage and bond formation. They sometimes will include a cofactor, as we will discuss, that encourages that bond breakage or bond formation, or that encourages the active site to have a particular shape. But basically, this active site is a pocket where that has a very specific chemical makeup where the reaction can occur. And then you have the substrate. That could be 1, 2, 3 molecules. It depends upon what the enzyme is designed to do. Those substrates come in and they interact with that active site, again, causing the substrate's bonds to break, form, whatever is necessary. And then the substrate leaves, and another substrate will come in. And then leave, and then more substrates in and leave. These pockets, again, they have to have a very specific chemical makeup in order for it to cause the reactions to occur. And we'll see that more later in this particular chapter. enzymes will differ in their degree of specificity. We looked at trypsin just a few moments ago on the example that was on the board. And it specifically focused on two amino acids. Some are very specific as to what they can accomplish. For example, catalase. Catalase is an enzyme that the only thing it does, its specificity is that the only thing it does is destroy hydrogen peroxide molecules. Which is a good thing, since hydrogen peroxide molecules cause aging and deterioration and many other issues for the body. So again, some of these enzymes will differ in the degree of their specificity. Some, it won't matter what they work on. Some are going to be very specific as to what they work on. When we get into this specificity, it also, they have specificity related to the whether they can work on L materials or whether they work on D or R materials. They can't work on both. So again, some of these like catalase, it's very, very specific. It is only going to decompose hydrogen peroxide in the body before the peroxide damages the cells. Thrombin is specific. It focuses strictly on peptide bond adjacent to arginine. So if the specific peptide doesn't have an arginine, it's not going to work on it. It only focuses on the bond adjacent to arginine. Carboxypeptidase is less specific. It tends to remove many of the C terminals. So in other words, it's going to react on the far right-hand side and just remove a C terminal. And papain, again, less specific, because it'll hydrolyze peptide bonds in many locations. Trypsin, which we looked at, focused on two. So it's all dependent on, these enzymes are built for specific things. And sometimes they have a specificity associated with them.
We have to memorize these specific enzymes.
No. I say that. I expect you to know catalase.
Okay.
And we will talk about how some of these are named. And when we get into how they are named, then I'll expect you to understand what they do. But as far as these specific ones, the only one I expect you to know is catalase, because it has one job and it's an extremely important job. I'm trying to let those that are writing catch up with me. Again, when I talk about specificity, they not only have specific things that they can control, but they also are going to have specific stereochemistries they can control. You can't fit a rectangle in a triangular hole. If it doesn't have the exact right stereochemistry, then it's not going to go into that particular active site. Or if the active site doesn't have that particular stereochemistry, then the substrate can't fit in. So let me give you an example. If I were to do this particular compound compared to this compound. Let's talk about what needs to be present. Let's say the enzyme comes in here. Let's talk about what, and remember, enzymes are just big protein chains, right? You're going to want hydrogen bonding capability on the top, and you're going to want London dispersion on the bottom. That is true for both of these. But if the middle of this has hydrogen bonding, H bond London dispersion. If this is our enzyme, this will not occur. It will not occur because that does not match. In its type of properties. So again, the stereochemistry is going to be important. You're going to have it in a certain direction in order for it to match up. Because if the intermolecular forces do not match up, then it's not going to cause the reaction to occur. So there is specificity. There's specificity on the enzyme. There is specificity on the substrate. These things are geared to be very specific. Some are geared to be very specific. and some are geared to be less specific. But when we talk about specificity, we're not only talking about what type of compound comes in, but also the stereochemistry and the direction it comes in. I couldn't bring this in upside down, or this one in upside down. Because if I tried to turn it upside down to make it work, then the north and south wouldn't work. So it's not a matter of just flipping this upside down to make it work, because then it's even worse. Now, your catalysts are designed, again, there is some specificity to the design of the catalyst. And that specificity also can include the turnover rate. How fast are the substrates going to enter, react, exit? That's what we mean by turnover rate. How fast are the substrates going to enter the enzyme, undergo the reaction, and exit the enzyme? Some, like papain, which hydrolyzes peptide bonds and does not have a specific peptide bond it hydrolyzes. It can only take in 10 peptides per second. Whereas thankfully, catalase takes in 10 billion peroxides per second. So basically, these peroxide, it's a industrial workforce. In, out, in, out, in, out. It gets the job done very quickly. And no, you don't have to memorize these. You need to understand turnover rate. What does turnover rate mean? And if I were to give you a turnover rate, what does that mean? I'm not asking you to memorize that catalase is 10 to the 7th. I will tell you catalase is 10 to the 7th. What does that mean for its turnover? What is, or I'll ask you to define turnover rate. What does turnover rate mean? I'm not asking you to memorize all of these. Questions? So far, what have I talked about that's important? I've talked about what an enzyme is. An enzyme is a catalyst that catalyzes specific reactions. Some enzymes are more specific to what they will catalyze than other enzymes. Catalase is one of the most specific because it will only do one job. That's the destruction of hydrogen peroxide. These enzymes have a specificity to them. They have an active site, and if the substrate does not line up with the active site, it will not work. Whether that is because of the type of intermolecular force, or whether that is because it's just the wrong shape of molecule. Enzymes have a specificity about them. Stereochemistry plays a role in that specificity. If you alter the stereochemistry, you can alter whether the enzyme will actually work. Enzymes have turnover rates. How fast can they turn over the substrates? Catalase, thankfully, is it's the fastest of all the enzymes. But a turnover rate is how fast does the substrate come in, undergo reaction, and exit. Now, I mentioned this once before. Sometimes these enzymes have to have cofactors. These cofactors help in shaping that active site, or they can help in causing the reaction. These cofactors can be a metal ion, hence the reason your vitamins are important. We know we have to have so much zinc. We have to have so much magnesium, manganese, you name it. Some of that is because they are cofactors to these enzymes. These cofactors can also be other non-protein organic molecules. But again, what the cofactor does is it does potentially a multitude of things. Number one, but the main thing it does is it causes the active site to have the right shape. Whether it is in the active site itself or whether it is added somewhere else in the enzyme to cause the active site to have the correct shape. Some of these enzyme cofactors are important because without them, the molecule would not be able to catalyze the reaction unless the perfect functional groups were present. It kind of gives that substance to help with the functional groups. For example, I keep mentioning catalase. Catalase requires iron 2. Without iron 2, catalase is not going to do its job. It's important to have magnesium because otherwise the hexokinase, which it destroys the glucose 6-phosphate in the glycolysis chain, isn't going to do its job. So these cofactors are important. They're typically your vitamins or trace minerals that we get from vitamins. And again, it helps in creating that active site. Perhaps these particular amino acids would not line up in this particular shape if the zinc 2+ wasn't there reacting with all these lone pairs on those side chains. This is an example where the enzyme cofactor is a mineral. But you are going to see as we get into the glycolysis chain, the citric acid cycle, and the electron transport chain, you're going to encounter one that is an organic compound. An example would be NAD plus, nicotinamide adenine dinucleotide, or perhaps it's coenzyme A. Those are examples of organic molecules that react as coenzymes that help cause the reaction to occur. Now this starts probably the hardest part in this chapter. This begins the part where, yes, you're going to need to know the terminology. Because this terminology is used to tell us what goes on in a reaction as an enzyme. This is enzyme classification. The first class we're going to talk about are oxidoreductase. Oxidoreductase has the name oxidation and reduction. So oxidoreductase causes either oxidation or reduction. That is the class, oxidoreductase. There are 3 subclasses of oxidoreductase. The first subclass of oxidoreductase is an oxidase. When you hear the name of an enzyme and it ends in oxidase, it means that it added oxygen to cause oxidation. If you hear reductase at the end of an enzyme name, it means that it added hydrogen, typically, to reduce the substrate. And then there's dehydrogenases. Dehydrogenases remove 2 hydrogens to form a double bond. So this would be an example of a ethyl dehydrogenase. Because or NAD plus would be an example of an ethyl dehydrogenase. Or ethanol dehydrogenase. It took that OH, it removed a hydrogen from the OH, and it removed a hydrogen from the CH2 to make a C double bond OH, an aldehyde. In so doing, it reduced the NAD plus to NADH, and you have an H plus. So this is a dehydrogenase, removal of hydrogen. A reductase reduces the substrate, typically by adding hydrogen. An oxidase oxidizes the substrate by adding oxygen. So how would this be asked on a test? I could say a dehydrogenase was acted upon this molecule. or NAD plus as was added as a dehydrogenase to act upon this molecule, what is the product? By the way, if you would like to see this in a table, table 19.4 on page 596 and 597, give you all of the enzyme classes and subclasses. The next class is the transferase. Just like its name says, it transfers something. In this case, it's a transfer of functional groups, but it transfers something. There are two subclasses to the transferase. The first subclass to the transferase is a transaminase. Amine, amino. It's telling you what it transfers. It'll transfer an amino group. Now, one that is not as specific as far as you have to know it because it doesn't tell you what it does immediately in the name is a kinase. But a kinase is an extremely important enzyme. Kinases are actually how we get our energy from, how we get the energy. They take place in the glycolysis cycle or the citric acid cycle. You're going to run into kinases in the electron transport chain as well. A kinase is something that transfers a phosphoryl group, a phosphate. A kinase transfers basically a phosphate group. Kinases is is ADP and ATP. ATP to ADP means that a phosphofructokinase, in this case, it's a phosphofructokinase. Phospho meaning it transfers a phosphate group. Fructo meaning it transferred it on fructose, a sugar, and kinase is telling you it's transferring that phosphate group. So these names can be very explicit. And I'm trying to let people catch up with me. Try not to go too fast. So again, if I say, something transaminase, it means I'm going to be transferring an amino acid to the substrate or between substrates. If you add the word kinase, it means you're transferring some sort of phosphoryl group. You need to be prepared to look at a set of molecules and identify what got transferred and tell me what the class and subclass is. I know for a fact that is one of the sets of questions. I will give you a series of reactions and I ask you what's the class and subclass. So you have to understand what these words mean. And not just the reactions that are shown here, but I can give you any reaction. You have to understand what the words mean. The next class is a hydrolase. Hydrolase. We've heard hydro before in hydrolysis. And that is what we are dealing with, is hydrolysis. There are 3 subclasses to the hydrolases. The first subclass is a lipase, lipid. So in other words, it hydrolyzes a lipid bond, an ester bond. The second subclass is a protease. Protease. Means it hydrolyzes a peptide bond. So in other words, trypsin that I used earlier is an example of a protease. Papain was an example of a protease because they hydrolyze a protein, a peptide bond in a protein. Nuclease. Nuclease specifically focuses on nucleic acids. RNA, DNA, nucleic acids. Specifically, it hydrolyzes the phosphate ester bond in a nucleic acid. So in other words, it's going to pull off a phosphate in a nucleic acid. Kinase, don't confuse it with kinases. Kinases, yes, they transfer a phosphate group. But a nuclease hydrolyzes, removes a phosphate group specifically from a nucleic acid. Yes.
Just to clarify, as far as the test questions go, we're not completing the reactions, we're just recognizing what's going on in the reactions.
Correct. Because completing the reactions would be quite challenging at this point,
Right.
not knowing where things should go. So it's more about recognizing what is going on.
Oh,
And then in this case, recognizing what is going on and then figuring out what class and subclass is associated with it.
oh.
Example would be 1942 and 1943. 1946. Those are some examples where you're specifically given a reaction and you're asked, what enzyme would you expect to catalyze it? Okay, the next one does not have a subclass. It just has a class. And it tells you what's going on. Again, folks, these words, if you understand the words, they're telling you what to do to it. Isomerase. I'm going to be doing I'm going to be going from one isomer to the next isomer. Basically, in this particular case, I've transferred a hydroxide group and a double bond. Visually speaking. In reality, what happened is this hydrogen went up here, making a hydroxide group, and this hydrogen had to attach to the carbon, and ultimately you made a double bond. You will notice that these two molecules have the exact same chemical formula. They're just arranged differently, hence isomerization, and hence isomerase. Besides your amino acids, table 19.4 is the table I would start studying hard for the next test. Because I think, again, this is amino acids was the challenging part in chapter 18. I believe this is the challenging part of chapter 19. Table 19.4, which is all of these classes and subclasses. The next class has 4 subclasses. It's the lyase. Basically, you'll notice it catalyzes the addition of water, carbon dioxide, ammonia to a double bond. And I'm missing one. The subclasses in your book are slightly different. So you do have the decarboxylase, carboxyl, carbonate, or carbon dioxide. It's telling you you're going to replace a carboxyl group with a hydrogen. Basically, you're removing the carbon dioxide. Dehydrase. You're removing water from a substrate to give a double bond. In your textbook, it's different than what I have. We talked about decarboxylase, that's the same. There is a deaminase. Which removes an ammonia. There is a dehydratase. Which is removal of water. And there is a hydratase. Which is the addition of water. Those are actually the 4 subclasses of liases. Folks, again, now, do not confuse dehydratase with dehydrogenase or dehydrogenase. Don't confuse dehydratase with dehydrogenase. It's easy to do. But if you look at the difference in the words, dehydrogenase has hydrogen in it. It's removal of hydrogen. Whereas dehydratase does not have all of the word hydrogen in it. And it's removal of water. So what is fumarase an example of? It doesn't tell you what it is. So what is it an example of? It's an example of hydratase because it added water. It is 9:15. We'll finish up this and move on to how enzymes work and I'll do some examples of how to determine what's going on. I'll do some examples and then we'll move on to how enzymes work on Tuesday.
Oh, yeah, he said she was.
Yeah.
For what we're doing now. Just-
I think I said, yeah. I have like 7. Okay.